lecture is moving on to talk about biotechnology. If we look at the word itself, we should be able to recognize some parts of the word. Of course, we know that bio has to do with life. And technology, this is an interesting word. We use the word a lot, but we may not actually be thinking about what the word technology means. Usually technology means something that can do a job better. And so when we look at biotechnology, the definition from the United Nations is any technological application that uses biological systems, living organisms, or derivatives thereof to make or modify products or processes for specific use. So what we want to pay attention here to is to make or modify products or processes. So what does that mean? What does that have to do with living things? The products or processes of living things. That's what we really mean. The products or processes of living organisms. Usually, specifically, a protein. Okay, we can talk about, for example, genetically modified organisms that can perhaps produce larger amounts of carbohydrates. But keep in mind, when we think about what in a plant actually produces carbohydrates, it's the process photosynthesis. That requires embedded proteins. So really, biotechnology is going to have a lot to do with genes and their products. So the product is a protein, and then the gene itself. And so that goes back to DNA. Biotechnology is mostly impacting DNA. So what might we want to do with DNA? It was interesting, we hear conversations when you all are talking about siRNA, we hear the idea that we might want to modify it. Why would you modify DNA? Right? If we want to modify it or we want to change it, why would we want to do that? Well, sometimes we might do that to correct a mutation. So if we're talking about a human who is living with cystic fibrosis, perhaps we want to correct that mutation. I heard someone else saying, well, maybe it's not a mutation, but maybe we just want to change the product. Maybe we don't want the child to be brown haired, maybe we want them to be blonde haired. And so we might want to change the phenotype of a trait. So that would be of a given trait. And so we could say, for example, hair color. But there's something else that we might want to do with DNA. And some of these are going to be really extreme and will never happen, but others might not be quite so extreme. And this might be to add traits. So some people will think about, like, oh, can you make me, could a human ever have wings? Well, remember, wings are going to be the result of many, many, many genes. So the likelihood that we could genetically engineer or use biotechnology to change that DNA is very unlikely. But for example, pesticide resistance. There are some plants that have a natural resistance to a chemical. There are other plants that if we took that gene, we could add that gene to other plants. So the idea of adding traits to an organism. So we could correct a mutation in a gene. We could change the phenotype of a given trait or a given gene. Or we could actually add entirely new genes or traits to an organism's genome. But the question is, how do we actually do that? It sounds really easy, but we know that it's not. We know that in able to access the DNA, remember the DNA is in every cell. And the DNA, especially in eukaryotes, surrounding it. And so one of the things that we have to think about is how complex it's going to be to actually be able to manipulate genes and manipulate proteins, to make these changes. However, there are certain tools that we already have if we want to change DNA. So for example, think about how DNA replication occurs and how we can proofread DNA. What is it that does the DNA proofreading? Well, DNA polymerase can proofread. It's able to make small changes in the DNA sequence, but we actually have a whole set of enzymes that are able to dramatically cut out sections of DNA. And these are called endonucleases, or you've probably heard them called restriction enzymes. Restriction enzymes are actually able to cut DNA. Cut or cleave means the same thing, DNA. And so the picture that you drew on the note card was actually a restriction enzyme cutting DNA at a specific location. What we need to be careful about is it's not just enough to be able to cut the DNA. We actually need to be able to deliver DNA. If I'm going to put new DNA in there, I not only have to cut out the old piece that I don't want, or cut open to be able to get in there, but I also need to be able to deliver the DNA. And we actually have two things. We were just talking about one, HIV, which delivers nucleic acids, and those are viruses. And then actually plasmids. We're going to talk a lot about those little extra chromosomal pieces of DNA in bacteria because they can switch and deliver DNA to each other. And so viruses and plasmids are what we call vectors. They deliver DNA. Okay, so I can cut DNA. I can take something out, or I can choose something that I want. I can deliver it into the cell and possibly into the nucleus, but I can't just have pieces of DNA. I need to seal it back together. So remember, we have something that allows us to seal DNA, seal or connect DNA, and that's an enzyme called DNA ligase. And we talked about that a lot when we talked about Okazaki fragments and how we need to connect all of those Okazaki fragments. So in order to truly understand biotechnology, we have to really make sure that we're thinking about what are the possibilities? What could we do to DNA? What tools do we have, are we going to need in order to be able to do this? Remember, here we're talking about mostly enzymes. That's not something you can necessarily build in a lab. You need a gene to make a protein to make an enzyme. But some of these things do exist already in cells, and we can use them or take advantage of them. So in order to perform biotechnology, you really need to know these tools. These are the tools that already exist that we're going to be using. There are some current examples of biotechnology that we're going to be focusing on. The first is going to be gene isolation, gene and protein isolation, which is how we find genes. So when we're talking about, oh, that gene is involved in Alzheimer's, we need to be able to find and isolate that gene. We are also able to perform what's called genetic engineering. Which requires many steps. It requires us to find a desired gene, deliver the desired gene to a cell, and what's called incorporate the gene. Incorporate the gene into the host genome. not just enough. You won't ever find genetic engineering where they say, okay, here's a drink or here's a shot. Now we've changed your genome. We have to actually get it into the cells and into the main part of the genome. And so genetic engineering, when we talk about genetic engineering, we're going to really focus on two main examples. And these are going to be bacteria. For many reasons, bacteria are the easiest to genetically manipulate. A, they have a small genome. B, no nucleus. And C, no post-transcriptional modifications. So these are all differences that you should already know. And then we're also going to see a lot more genetic engineering on plants. People, of course, don't consider plants to be as alive, per se, as animals. So if we talked about genetically engineering an animal, people would have a lot of objections. But when we talk about genetically engineering plants, a lot of the food that you eat is already genetically modified. The final type of current biotech is what's called gene therapy. And gene therapy is not as successful as genetic engineering. This is where we are going to be replacing defective genes. So if there's someone who has cystic fibrosis, can we replace that defective gene with a functional gene? These are the three main examples of biotech. What we're going to be talking about for the rest of this lecture and the next lecture is how are these actually done. First, we're going to focus on restriction endonucleases, right? Restriction enzymes, and their ability to separate and identify segments of DNA. So the first thing that we need to know is that restriction endonucleases are actually able to, and if you can read this, it'll talk you through it a little bit as well. Restriction enzymes are actually going to be able to cleave the DNA at specific sites. There are many restriction enzymes, many different sites, many unique sites. So that's one of the first big things that we need to know, is that restriction enzymes are able to cleave or cut DNA at specific sites. They're actually able to cut it into what are called blunt or sticky ends, meaning that they're able to leave an end that we can add something onto, but we'll get into that a little bit more later. One of the things that we need to focus on is that once we actually cut it, we can separate the pieces. So we're going to focus on a process called gel electrophoresis. Gel electrophoresis is where after it's cut, you add electricity. And this is going to push the segments through a gel. And this gel is going to separate by size. So longer pieces are not going to move as quickly. Shorter pieces are going to move further. So longer pieces are heavier, and the electricity doesn't push them as much. Now we do need to understand that the electricity is actually separated. Electricity comes in negative and positive ends. So the negative end is actually going to be placed where the DNA is placed, or where the DNA is originally placed, because DNA is also negatively charged, and negative charges repel. So if I have my negatively charged DNA, and I put a negative charge there, it's actually going to repel the DNA away, push it through this gel. The different parts of the gel will actually separate it, and that's how we get what's called a DNA fingerprint or a gel electrophoresis. From there, you're actually able to pull those pieces of DNA out using buffers and these nitrocellulose pads, and that's how we're able to actually grab pieces of DNA. This is where we're going to stop for now. Please make sure that you read over the steps that are listed there.